Center Grand Rounds. We have kind of a special presentation today. As some of you may be aware, there's really quite a remarkable resource that's been developed on the University of Colorado Anschutz campus over the past several years, brought under the umbrella of the Colorado Center for Personalized Medicine. And it's really come a long way. I've had the pleasure of sort of watching from a distance as this has evolved. It's really come a long way in the past few years, and I think represents potentially a extremely powerful resource and interaction with the Cancer Center. And so since we haven't had, at least in recent times, haven't had a whole lot of interaction with this group, we thought it would be quite a good time to have some of the representatives of that program come and speak to the Cancer Center community more broadly. And so I really appreciate, we have not one but three members of the CCPM team that are here today. And so let me just briefly introduce them. And I think they're going to sort of tag team for their presentation, which will cover some of the resources that they're providing as well as some of the research they're doing. So I believe first off here we have Dr. Nikita Pozdeyev. Nikita is an MD PhD who was recruited to the University of Colorado just last year to be the director of the service they're going to tell you about. It's called the Translational Informatics Service for the CCPM. And so he is a cancer investigator himself with a focus on thyroid cancer and collaborates with several of the groups on campus already. And so as a Cancer Center member, so already working with that, and we're excited to hear more about the programs that he's developing. He's also going to be joined by Nicolas Rafaels, who's the Associate Director of the Translation Informatics Services. Nicolas has a background in mathematics and biostatistics and has many years working on complex studies in genetics research, as well as helping develop some of the computational pipelines that the CCPM uses. And then lastly, we have Professor Ethan Lange, who is a statistical geneticist with expertise in the development and application of different statistical methods, has worked on a wide range of studies on multiple diseases, including cancer, and his particular interest is prostate cancer. And he has also a background in biostatistics and applied mathematics. So gentlemen, again, really appreciate all of you being here today and look forward to hearing more about the programs that you're developing. So Nikita, you're welcome to take it away. And Jill, if you're not recording, please do.
Thank you, Craig. It's an honor and a pleasure to tell you all about the service that we have created and also about the resources that we have in CCPM and Translation Informatics Services. As you know, the CCPM Biobank consents, recruits patients, and collects blood samples and genotypes, patients who are treated at UC Health facilities. And Translation Informatics Service, which has been created just 6 months ago, is an operational unit with a primary goal to work with this genetic data and make it available for researchers at the university and beyond. So in this talk, for the first 15 minutes, I hope to tell a little bit about what we are doing in TIS and what kind of services we can provide for you. Review data that we have and the analysis that we perform. And Nick is going to go deeper into the details of this analysis. In preparation for this talk and also in our earlier discussions with Craig and Jennifer, we pulled some data on cancer cohorts as well as some pathogenic germline variants in the biobank genetic data preset we have. And finally, Eden will tell about interests of CCPM and DIPM faculty as well as cancer-related projects that he has been working on. So in Translation Informatics Services, we have three main missions. The first one is we support CCPM operations, such as delivery of genetic data for clinicians in EPIC electronic health records. We also take raw genetic data, perform quality control, and convert it into a usable data sets. And finally, especially in the last 2 years, CCPM has been developing partnerships with industry, and we support these partnerships as informatics core. We extensively work with researchers from within CCPM and also from the other divisions and departments in the university. There are two main services we do. First is a cohort discovery. If somebody is interested in what we have in our data sets, we can provide this information. But more importantly, we run data analysis pipelines, mostly genome-wide association studies, and we have two state-of-the-art pipelines that are called Sage and Regini that we currently are capable of running, and slowly getting into the polygenic rescourse space in collaboration with CCPM and PIPM faculty. And finally, our third goal is to make the data available and work with researchers who could use this data to create new hypotheses, apply for grants. As I mentioned, we are able to deliver this data directly to researchers and also results of the analysis that we do. But we also develop software that makes genomic data exploration possible even for investigators with no informatics experience or programming experience. We also hope to provide education in those areas where we work. So on this slide, I show main sources of the data that we have. And primary, this has been CCPM Biobank. And as of January 4th this year, there are 168,000 consented biobank participants. For 72,000 of these participants, we already have samples collected, and 33.8 thousand we have genotyping data available. You may have heard of CCPM, a collaboration with Regeneron Genetics Center. The great benefit we'll get from this collaboration is we'll get whole exome sequencing on biobank participant samples. 10,000 samples currently are being genotyped by RGC. There is a second batch coming of 70,000. But our ultimate goal in 5 years to have almost 500,000 genotyped UC Health patients. Our biobank is not the biggest. It's actually more on the smaller side compared to the big initiatives such as UK Biobank or Million Veterans programs. But there is a big strength, which is in the clinical data. Each of the genotypes that we have is linked to the detailed clinical data that has been extracted by Health Data Compass from EPIC EHR. And we do have access to this clinical data for biobank participants. We use so-called P codes, which is an aggregated version of billion ICD codes as our primary phenotype definition. Those have been calculated by Dr. Wali lab, but we also have some other data such as labs, medications, procedures. So you could get really creative with cohorts that we could identify and analyze. And importantly, we also work with Cargo, and Cargo is a web lab that performs cheap and targeted genotyping, as well as methylome analysis. We serve as an informatics core for Cargo, and if an investigator is interested in bringing a different cohort of patients who has not been represented yet in our data sets, such as, for example, a specific cancer cohort, then by working through Cargo, we can process, genotype these samples and also analyze them and make them useful. So because of interest in exploring underserved and underrepresented minorities in Cancer Center, we pull this data, which are counts for different race and ethnicities in genotype biobank data freeze. not surprisingly, most of biobank participants are white, non-Hispanic. We have about 1,400 Blacks and 2,700 Hispanic patients as well, who have been genotyped already. For request of Craig, we looked at 4 common cancers and also looked at how many Blacks and Hispanics we have. Numbers are not that high, which will limit what we could do, what kind of analysis we could do. But these numbers are expected to grow, and those projections in 5 years, when we have 450,000 biobank participants, we expect to have hundreds of cancer patients from Black and Hispanic minorities. I'd like to briefly mention our effort on making this data easily explorable. This is the portal that we have developed for the Asthma Kappa Consortium, which allows to query genome-wide association studies, summary statistics in many different ways by gene, transcript, variant. And we could do this for the cancer-associated phenotypes as well. A big initiative that we are just starting is Colorado Biobank Engine, where we will have hundreds of phenotypes available through this graphics user interface portal, which will obviously include many cancers. And that will be available to university researchers. I'll briefly mention Sue Fang's workshop that will be available in a couple of months for those who are interested in diving deep and learning how this GWAS pipelines are actually working. So importantly, should any of you be interested in working with us, how do you get there? And for a simple cohort request, this URL will take you to the survey. It's a very simple, easy to fill the survey, which will tell us what cohorts you are interested in, and we'll pull this out of our databases. For bigger projects, there is a formal approval required. Brianna Mayfield, she is our project manager, and she's also a coordinator for the Access to Biobank Committee, and she can guide you to this process. It looks complicated, but it's actually not that onerous. And then we could do a real large-scale research together. And I will post in the chat window all these URLs. So the final slide that I have shows Translation Informatics Service in its full glory and some fun facts about our work. We are very collaborative, looking forward to potential collaborations. And on this note, I will let Nick present his slides.
Great, thank you, Nikita, and thank you, Craig, for a wonderful introduction. Try and share my screen real quick. Just bear with me. Did everyone see my screen? I'll take that as a yes.
Yeah,
so what I wanted to do, thank you. What I wanted to do first was just do a brief background on the type of data that we have in the biobank and why we decided to go that route and how that can help you. So I'm not sure if everyone is familiar with what is a GWAS and how we do imputation. So basically, these chips are designed such that we only select the most common variants that are found in about 1% in the population or more. And so instead of having to sequence all 3 billion base pairs, we can get away with genotyping a couple million base pairs. And so you see we select these haplotype tagging variants here that are representative of the full haplotype background. But then we can recover that information by doing imputation, by matching it to a reference haplotype where the rest of these variants are known given given the variants that this individual has, and then you can fill that information back in. So why do a GWAS? Well, mostly because of cost efficiency. So on a per sample basis, the mega genotyping which is the panel that we're using in CCPM, is 2.5 to 12 times less expensive than doing any of these other methods like whole exome sequencing or whole genome sequencing. And so there's a great benefit in terms of cost and trying to get this out to, tens or hundreds of thousands of patients. And so, but well, what are we losing when we go down to a mega versus whole genome sequencing? Well, not as much as you might think. So we're able to actually capture a lot of these interesting variants that may have clinical utility on the mega, and we can also design custom content as well to kind of capture even more information. So for example, on our latest mega chip, we were able to capture almost all of our PGX content, the hemochromatosis and factor V Leiden we're able to capture, and more than 50% of all the variants that are found in ACMG. And so the mega can predict approximately 92% of comparable actionable genetic variation identified by whole exome sequencing at less than 10% of the cost. So as you might find in cancer research, but it's also true in doing genetic research, is that there's a European bias in GWAS studies. And so as we see, and there's a paper here from Alicia Martin a couple of years ago, even over time, while we are increasing the numbers of doing GWAS in minority populations, we're still getting the bulk of our information in European ancestry. And so this certainly can have an effect on how well we're able to, I guess, deal with ethnic disparities in susceptibility and survival. As you see from this figure in the Alzheimer review paper, genetic polymorphisms is one factor in different factors that contribute to susceptibility and survival disparities in cancer. So how do we deal with this? Well, one part is to develop this mega chip. And so back in 2015, some future collaborators of future faculty of CCPM, Kathleen Barnes and Christian Yu, teamed up to develop this mega chip. So Dr. Barnes had done whole genome sequencing on over 900 individuals from 21 sites in the Americas and a couple in Africa, all of African ancestry, to uncover a lot of variation that hadn't yet been known. In addition, through PAGE, they did 36,000 whole exome sequences from Latino and African populations and uncovered a lot of new exome variants that hadn't yet been discovered to kind of contribute to clinical utility. And so this is kind of where we started as a baseline when CCPM was started. Another thing that can help us deal with some of these ethnic disparities is to develop a better imputation panel. So TopMed, which is this NHLBI study focusing on whole genome sequencing of over 100,000 subjects. And they try to target populations with rich phenotypes and diverse populations. And so the key here is the diverse populations, at least for our benefit. So a lot of these populations are of African and Latino ancestry, which hadn't really been whole genome sequenced to a large degree. Like I think Kappa might have been one of the biggest ones before this. And you can see here on the left the improvement in imputation on this. These are on the top here is an African ancestry panel from Jackson Hart. And I think this is a Hispanic population, but you can see that compared to the commonly used imputation panels previously, 1,000 Genomes and the Haplotype Reference Consortium, we do see quite a bit improvement, especially in African ancestry populations, in imputation quality than we were having before. And so we can see this in these numbers here. The coverage is 1.82 times greater in African populations compared to 1,000 Genomes for TopMed. And so what's also cool about imputation is that you can actually uncover a lot of functional variants that you hadn't yet genotyped before. And so here, for example, we have found 23 loss of function variants in the BRCA, one of the BRCA genes. And I guess this is among 12,636 cases of cancer. And so that's something that's exciting that we wouldn't have been able to do otherwise through just genotyping. And then finally, because we have such a large reference panel, we can impute to a very, very fine degree of, for example, in this study, 1 out of 10,000 patients, we can find quality imputation at that level, which is really unprecedented. So kind of going back to what Nikita was talking about. So here's an overview of kind of where we are with our biobank freezes and where we want to go. And so, like Nikita was saying, we get our data from the biobank, we do some processing on that data. And so we process that data in 2 iterations. We had our initial mega chip, which we really, the custom content there was just asking PIs on the Anschutz campus, like, hey, what genes or SNPs are you interested in? We thought, well, then after a year or two, we realized that actually we want to kind of add just a bunch of really good clinical content. So we use the expertise within CCPM and BIPM to kind of add a lot more, pharmacogenetic and clinical SNPs. And so that's what was added the second time. We do some processing, and then after some quality control, these are the final numbers that we get. And then we run that through the TopMed imputation server. And so now we're able to get, from these less than 2 million variants, we're able to recover 50 million variants, which is really exciting. And then in the future, we hope to, get our data back from Regeneron. And when we do that, we'll not only get the whole exome sequencing data, but we'll also get the global screening array data. And so that can kind of fill in the gaps in the intronic and intergenic regions. And so we can also repeat imputation and kind of add that to our pool of data that's available for research, and kind of pool this data, which doesn't have exome sequence data yet, with some data that does, and kind of work with all of it together. So we are ready to take your GWAS requests. So thanks to some work from Tanya Brunetti, we've created a biobank GWAS pipeline that you can actually download and use yourself, but you don't have to. We're happy to do this for you. So this, again, uses the SAGE analysis pipeline, and really the benefits of SAGE are, it deals with, I guess, relatedness within the data, also the various ethnic groups, and also deals with unbalanced case control design. So it uses some really nice statistical models to kind of handle that really well. And so as we see here, we have so a previous association we did for validation, we find that we are able to replicate some of the breast cancer signals running the GWAS here. I think an important thing to note here of how we want to do analysis for, with the Cancer Center in the future. I think, the cancer, I think, has many different etiological backgrounds. And so, in some respects, we can kind of look at the rare variants, and they're more monogenic in nature, and so Mendelian, and so they have larger effect sizes. And so that's stuff we can look at in ClinVar and pathogenic variants there. But then there's also a bunch of common variants which may be associated in some of these more heterogeneous diseases and common cancers. And so so that's discovered from GWAS. And so we'll talk about additional methods we can use on top of GWAS to kind of help uncover all these common variants. And as you see in our biobank, and this is true and across all of human nature, is that for the most part, most of the variation that we find is really, really rare. And there are very few common variants, as I was talking about with the imputation, only a couple million variants. So getting back to rare variants, and so what we wanted to do was take a look at our biobank, look at all the pathogenic variants in the most recent release of ClinVar, which was a couple weeks ago, and see what, if there are any differences, or between ethnic groups for any of these variants. And so we looked at the African American, non-Hispanic, and any Hispanic versus European, and we ran some Fisher's exact tests and did a Benjamin E. Hochberg, I guess, multiple comparison check to see if we found anything that was of interest. And so unfortunately, we can't show any actual counts because these counts are all very small. We can't reveal anything less than 10. But what I can tell you is that we did find an association, this ATM gene and a BRCA2 gene, both shown to be associated with breast and ovarian cancer syndrome and hereditary cancer syndromes. One such SNP was highly associated with Latin Americans, another one highly associated with African Americans. Basically, that all these variants are only found in this ethnic group and not in the other ethnic group. So in the future, what we'd like to do is similar to that TopMed paper, validate additional imputed ClinVar variants, And then of course, maybe how we can help this group is to find de novo variants from whole exome sequencing and discover new variants associated with cancer. So as I was talking about with the more common variants, I think the next phase that we want to go is look into polygenic risk scores. And so again, I think what we want to be able to do is take all of these common variants across the entire genome and then kind of determine like, well, what is the heritability across all of these variants, right? Because any individual common variant is actually going to have very, very low heritability. If we're able to find some way to kind of collapse all that information and kind of pool it all together into some kind of score, we actually can maybe potentially use that information to better classify like what is someone's risk of developing a disease and potentially use that in the clinic after some clinical trials. And so that's kind of the things that we're thinking about doing at CCPM, over time. And so here, for example, if you want to think about the way that we represent polygenic risk scores is kind of area under the curve. And so you think on average, most folks would have an 11% average risk in a population of this particular disease here. But then, if you fall under the curve to the left of that, then you have lower polygenic risk score, and if you fall to the right, then you have higher polygenic risk score. So some more background on how this works. So basically, you would run a GWAS, you get your summary statistics, but then of course you run a GWAS on a whole bunch of variants. Many of these variants can have high correlation that's represented by linkage disequilibrium, and then some of these other variants may have no contribution to that particular disease, even if they aren't an LD. So you kind of want to do some pruning of the set of GWAS statistics that you have to kind of get that final set of variants that you're interested in. And so once you're able to do that, and so you do some, and there's various ways and algorithms that people do use this, and then you would develop your training set on a separate population, kind of develop, pick out your best score, and then eventually you would then run that validation on your data, samples of interest, and then you would generate your polygenic risk scores. So finally, I think it's also important, just as another thing to deal with ethnic disparities is you have to be very careful about how you run your polygenic risk scores. In that same Martin paper, they show that if you develop your polygenic risk score on a totally European model, and you try to map that back onto other populations, it doesn't perform as well, and it performs extremely poorly on African populations. And so you need to adjust for that properly. And so here I'm highlighting on the right some work that Chris Earhart, also from CCPM, has worked on for atrial fibrillation, but this concept can be applied to any phenotype. And so you can see when he runs his models that there are different polygenic risk scores for different populations. While some are more similar than others, you can see that the African population is quite a bit off compared to Europeans. And so almost all African Americans would be considered to be at risk of atrial fibrillation, when in fact that's not true. And so you need to standardize this to some degree, so you can probably capture the more correct polygenic risk. All right, and with that, I will hand it over to Ethan, and thank you for your time.
Ethan, would you like to pause for questions now on the first part, or press through to the end? Happy to do whatever you prefer.
I'm fine with that. I know there's probably a lot of questions, so I think, yeah, let's go ahead with the
Okay.
questions.
Yeah, we've had a lively discussion in the chat box here.
Okay.
If I could throw in a question then. So Nick, thank you. This is a fascinating resource. And I was pretty excited that I know the numbers were kind of small, but you actually found some the BRCA variants in the African American population, which is encouraging that even at this stage we can begin to detect it. I think you told me, maybe you can elaborate, I think either you or Ethan had mentioned to me that polygenic risk scores you can calculate with smaller numbers. Is that correct? And is that the more sort of appealing thing to do at this stage of the development of the database?
Yeah. So yeah, Ethan did make that point, and I do agree that basically you don't have the power to find kind of these like single point genetic associations that you find in a GWAS study.
Okay. So at this stage of evolution, should people be focusing more on polygenic risk scores, perhaps?
I think my argument would be, I think we can focus on polygenic risk scores, but we can also focus on these pathogenic variants as well, because they would not be captured otherwise in the polygenic risk score to begin with, because they're so rare. You know, if we can uncover additional pathogenic variants through mutation and also through whole exome sequencing, that might give us two paths for us to go.
Okay, thank you. Other questions for Nikita or Nick? Okay, I think we can press on then. Ethan, it's all yours.
Great. I've been having some technical difficulties, so I'm gonna leave my camera off to spare some bandwidth. Hopefully that helps.
Okay.
And I'm gonna share my screen. Okay, that's not working. Sorry. More technical issues.
I can throw another question while you're working on that, Ethan. So as was discussed in the chat box, the current database is composed of normal blood specimens. So this is, for the cancer community, this is not tumor genotyping. And I guess the question I would throw out is, what is the appetite at CCPM right now for tackling tumor specimens? As you guys probably appreciate, there's quite a few tumor banks on this campus. And I suspect investigators that would love to have their tumors genotyped. Is that sort of within the purview or capacity of CCPM?
Yeah, that's a good question, Craig. So at this time, you're right. We don't do any somatic mutation profiling, and honestly, cheap genotyping technology is probably not the best one for somatic mutations, because you only look for a selected number of variants. It's difficult, not impossible, but difficult to do copy number variation changes. And also, you cannot detect gene fusions, which are probably the most clinically relevant variants in cancer. So it's difficult.
So
there are some, yes, go ahead, Craig.
I was just going to say, perhaps under the umbrella of the Regeneron collaboration, where they're doing whole exome sequencing?
Yeah, at this time, I'm not aware of any agreement with Regeneron to do whole exome sequencing on tumor samples.
Okay, so they're just interested in blood samples right now?
That's what the agreement is.
Okay.
For the germline variants.
Okay, thank you. Okay, I can see your slides now, Ethan, if you want to go ahead.
All right. So Nick and Nikita did a great job of sort of talking about the biobank resources. And so I thought I'd just quickly touch up on some of the personnel resources, some of the faculty research, because, obviously we're here to help. A lot of us have very relevant expertise, and we're eager to collaborate. Very briefly, the Division of Biomedical Informatics and Personalized Medicine, BIPM, was established in 2013. As of 2015, Kathleen Barnes was made the head of that. We have about 15 faculty research labs. Many of the faculty members have secondary appointments in other departments and schools, are actively participate in NIH and external support research projects, mostly obviously involved with genetic research, some microbiome, some genomics. Many faculty members have research areas that are focused on understudied minority populations, which I know lines up very well with the interests of the Cancer Center. Many of us are interested in studying underrepresented minority populations. And of course, faculty members are actively involved in student education as well. The Colorado Center for Personalized Medicine, which houses the CU Biobank Health Data Compass, computing resources, a bunch of the genetics cores and clinical operations, and of course, all the many exceptional personnel that make these operations run smoothly. Our faculty are scattered across the CU Anschutz Medical Campus, across the various schools, in addition to BIPM, as well as we have faculty in CU Boulder, UC Denver downtown campus, the VA, and National Jewish Health. So we really try to be an open group that brings discussion of all people interested in genetics across the genetic Denver area. Our center hosts weekly seminars, and it's open to anybody who's interested in attending. We have monthly faculty meetings and annual retreat, which are all excellent opportunities where we share ideas and build collaborative research. I spend about 5 minutes just sort of talking about my own research and how it relates to the Cancer Center objectives. My research is mostly, at the moment, focused on broad area of genetic and epigenetic risk factors for cardiorenal disease and associated biomarkers in large African American cohorts, including the Jackson Heart study and the REGARD study. Again, much of my research right now is interested in addressing this significant area of health disparity. And these studies include protein biomarkers, GWAS, PRS, sequence analyses, methylation, et cetera. And then I have methodological interest in improving efficiency of genetic study designs and polygenic risk scores. I have personally studied many different traits over my 30 years of working in genetics. I would say that if I had to choose one area that I focus most on, it's been prostate cancer. And that started when I was a graduate student at the University of Michigan, working with Kathleen Cooney, who is now chair at Duke, of medicine at Duke. This was originally a family-based study of inherited prostate cancer, involved linkage analyses, but we also, through the course of the study, collected anybody that came through the Michigan clinic who was diagnosed with prostate cancer prior to the age of 56. So hence we were able to collect a large number. We were a fairly specialized study in prostate cancer, focused on hereditary disease. This is an example of one of the families that we collected. And so very briefly, one of the things that we did was for the first decade or so that we worked together, we did a lot of family-based linkage studies. And during the course of that, we identified a strong linkage region on chromosome 17q, right near the BRCA1 gene. And we collaborated then with investigators of similar families at Johns Hopkins University, and we aggregated our data and found a strong linkage peak in this region, in which we then performed exome sequencing under that region. In 94 unrelated men from these families, we sequenced 2,000 exons under the 2-odd support interval. And during the course of that sequencing effort, we found one recurrent missense variant in 4 Caucasian cases, and then we found another missense variant in an African American case in a gene that had clear biological plausibility, namely HOXB13. Both the variants were predicted to be damaging by bioinformatic resources. HOXB13 is a homeobox transcription factor gene important in prostate development, and many mouse models have shown that knockouts of HOXB13 result in damage to the prostate. We also looked at external resources, so this is something we routinely do in genetic association studies. One such resource is the GTEx portal, which has done a lot of gene expression across many different tissue types. And what we found was the HOXB13 gene was primarily expressed in the prostate. So that was further evidence that it was an important gene. We then went and genotyped the remaining family members. We just sequenced the index case for this mutation, and we found that all prostate cancer cases in the families also harbored the mutations that we identified. We then went and sequenced 5,000 or genotyped 5,000 additional cases for this HOXB13 G84E variant that we found, and a number of screen controls. And we found that the variant was strongly associated with prostate cancer, and included to be so when we expanded our control population. One of the, another internal control was to see whether the HOXB13 mutation was more strongly associated in those who had prostate cancer. Was it more strongly associated with family history versus no family history? Presumably it's a high penetrant genetic variant. It should be associated with family history. It was. It was also associated with earlier age of diagnosis. And then combining those two was much more strongly associated in those that had family history and age of diagnosis prior to age 55. So this is sort of consistent with what you'd expect for a high penetrant mutation that we found. Some interest was the variant was primarily on a single Caucasian haplotype. It's the founder mutation that essentially was derived out of the Nordic countries. It's much more common in northern Germany and the Scandinavian countries. Our results have been replicated numerous times. It's probably over 20 times now. And the lifetime penetrance is somewhere around 50% for this mutation. And importantly, it is not independent of the burden of common variants, i.e. the PRS that we get from common variants influences that penetrance of that mutation. And I suspect that's also going to be true for many of the other mutations we find across cancer types, such as the BRCA mutations, that there too, the penetrance will likely be a function of the PRS. We also did a GWAS of 931 unrelated early onset cases. And really the purpose for me showing you this is, so we found one significant hit during our course of our GWAS with about 1,000 of these highly genetic prostate cancer cases. And we just found one significant hit, and that hit is a hit that was well known at the time. It's the 8q20, I believe it's 24, I can't remember, region that has strongest associated variants with not only prostate cancer, but also breast cancer and colon cancer. Some of these other peaks that we see in this area here are all, turns out to be all established prostate cancer loci as well. But this really gets to the point that you really need quite large numbers to do to have any kind of power to do a GWAS. So how do we improve on that? Well, it's polygenic risk scores. So GWASs have identified now tens of thousands of common low penetrant risk variants for hundreds of different traits. Most of these variants do not have a clear functional role. There's obviously an active area of research to sort of try to identify what the functional variants that underscore these GWAS signals are. And maybe among these, some of these functional variants may have some potential for providing mechanisms in new therapies. But a major area of focus in genetics research is how to use the broader GWAS discoveries to improve patient care, specifically in regards to targeted earlier screening and intervention strategies. And so for this, individual variants are not going to be very useful, aside from the small set of highly penetrant functional variants, such as we see in BRCA. But again, most GWAS variants are not going to have clear functional or clinical utility. They're not all that predictive. But when we find that when we aggregate data across hundreds of associated variants into these polygenic risk scores for a given disease, we can identify subsets of patients in the population whose risk for disease rivals those of individuals carrying high risk, rare high penetrant mutations. And so this is just another example with the prostate cancer research that we did. On the left here, you see a histogram of the distribution of number of risk alleles for cases and controls. The controls are the blue, the cases are the yellow, and the green is the overlap in the distributions. And what you see is in the prostate cancer cases, a clear shift in the number of risk alleles that these prostate cancer cases had compared to controls. One other way to look at that is to compare, look at the odds ratios when you split the data by deciles of distribution of total risk alleles in the controls. And what you find is that individuals that are in the highest decile have an odds ratio of approaching 10 compared to those individuals in the lowest decile for risk of prostate cancer. And so, there have been other studies with prostate cancer that show similar results. Maybe the odds ratio aren't quite as high. Again, these are pretty highly genetic cases that we looked at. But nonetheless, those odds ratios in the upper decile tend to be around 5, which for prostate cancer would be a significant risk factor, given how common it is. Here's another way of looking at it. This kind of an interesting thing was, this was a little bit of an early work I did, but the average number of risk alleles, and these are across 26 established prostate cancer variants. Now we have probably approaching 200 such variants. But what you can see is a clear trend line for those with prostate cancer. So this is looking at the cases, or the 4 bars on the left. The controls of the bar is the CGEMs there on the right. What you see is that the younger the prostate cancer cases, the more risk alleles they tend to have, which is consistent with the idea that genetic prostate cancer probably occurs earlier than non-genetic prostate cancer. So I'm going to go back now to the CU Biobank a little bit. So some of the strengths that we have at the Biobank are clearly we have multiple cancer types. We're not a singular focus study on a particular cancer. We have a large array of clinical data, including cardiovascular outcomes and other things that might be useful, especially as we think about treatment response to things such as chemotherapy. We do have ethnic diversity, and we have longitudinal follow-up. We're getting the blood samples from people coming into the clinic today. Presumably, they will stay in the health system, some of them for many years to come. There are obviously also limitations. We have a limited numbers per cancer type. The data is often messy, including controls. Our controls are not typically screened for the cancer types that we're interested in looking at. While we do have ethnic diversity, our population in Denver is mostly white, and the biobank reflects that. And there are likely high rates of loss to follow-up, people moving, dropping out, going to different hospitals, whatnot. So briefly about CU Biobank in terms of GWAS, as we talked about a little bit earlier here, there is a limitation for low power to detect novel genetic associations not already identified in whites, where there have been large consortia for many of these cancers that have already collected tens of thousands of cases. So while we may find significant GWAS hits in our own data, it's very unlikely that we're going to find anything new that hasn't been discovered previously. And then in terms of minorities where large GWASs have not largely been done, we're going to have low power because of our relatively low number of cases. And as I showed in that prostate cancer example, we typically require hundreds of thousands of cases to identify significant findings. With that being said, we should have decent power to replicate previous findings due to the lower significance threshold that we require when we're performing our statistical tests. And that will even probably prove true as our numbers get larger for some of the minority samples. There are many situations where for known genes that the variant that we find in whites is not the same variant that is important in the minority samples. It may be variants in the same region that are interesting, but they may be entirely different variants. And I've worked on several examples of that. So value can also be achieved in whites and minorities by including these data in large consortia. That's what many of us as faculty do. Our research projects now are largely collaborative, where we don't work just on our own individual cohort, but rather we work with many individuals across many cohorts and biobanks to build up our samples. And in this case, the CU Biobank will be a well appreciated addition to what is being done. And I think it would open opportunities for those who are interested to get involved in some of these consortia. And I think there's also value, particularly for rare sequence variants that we're obtaining through the future sequencing with Regeneron. Many fewer cases externally have been sequenced than versus GWAS arrays. And so there are some opportunities, I think, to find some new things for these low frequency variants that have not been well captured in GWAS studies. So genetic risk scores revisited. Again, individual associated variants have low predictive value, even if those frequencies vary significantly between populations, as they often do. The CU Biobank should be adequately powered to evaluate genetic risk scores. And it's got risk scores, genetic risk scores have many different names and acronyms, polygenic risk scores, PRS, GRS, just a few. Even in the smaller minority populations, which I think is important. And as Nick said, most of the PRs that we have today are developed in whites, and we need to assess their transferability and applicability in minority populations and work to improve them. And I think our biobank will be well positioned to do some of that alone, and also, I think, importantly, in collaborations with other groups. Sequence variants, low frequency variants remain, again, understudied in most cancers beyond established genes. And then even for some of the known genes, such as ATM, which I actually helped find 25 years ago, where many of those mutations are quite rare, and any additional information that we can have to characterize those mutations would be, I think, a considerable value to the research community. Many of the associated GWAS variants are shared across populations. That's largely true with some of these common variants. But high penetrant exome sequence variants are much less likely to be shared across populations. And so I think this is, again, another area where those who are interested in health disparity research, having large numbers of minority participants who have been sequenced, such as we're going to get eventually, will be of considerable value. I think other possible, another example of a possible use of the exome sequencing data is we can impute clonal I'm not going to just say CHIP, somatic mutations in the blood, which are strongly associated with hematologic malignancy and cardiovascular disease. There are other uses as well that the sequence data can do in terms of some ability to look at structural variants. And then there's some additional ideas. We can assess PRS for association with clinical features of disease, age of diagnosis, stage, et cetera, such as some of the examples I sort of showed with the age of diagnoses with prostate cancer. And I think those are kind of important. So we can sort of and then assess the role also of PRS in individuals carrying high penetrant mutations. By getting the exome sequence data, we're going to identify individuals with BRCA1 mutations, a number of them, for example. And we can see how much the PRS in those individuals influence the penetrance. We can also identify subsets of patients based on PRS and other genetic findings that could maybe perhaps benefit from earlier and more aggressive screening practices. I think there are many possibilities, and I think it would be great if the cancer researchers and us geneticists over on this end can put our heads together and discuss and come up with some neat ideas to pursue. And this is just some acknowledgement to the people who worked with the prostate cancer data myself.
Thank you, Ethan. Let's see, we have a few minutes here. We've had a couple of questions in the chat box. James was asking, what depth will the West be sequenced at?
I believe it'll be at least 20x coverage.
Okay. So Ethan, you hit on an issue that I was wondering about, and that is how readily, obviously there are some limitations to what we can do with our current database by virtue of its size. How readily available are some of these consortium interactions such that we could contemplate some of the questions that require larger data sets? Is that at a point where it can be sort of offered to the community, or is that still sort of an emerging sort of hit or miss kind of opportunity?
Well, so a lot of us are very well connected in the center with other outside cohorts. So, as our data, I think, is now getting finally polished and clean, at least the metadata is, I think we're now in a position where we can begin to actually reach out to some of these consortia, let them know that we exist and that we have these data. So I think that there are many opportunities that we probably can start really soon. Obviously, as we grow bigger, we're going to become a bigger and bigger player in those consortia, and to the point where it's possible we can even lead our own consortia. So yeah, I think there are opportunities there. There are some individuals we could reach out to. I know some individuals in the prostate cancer world. I'm sure there are people and others who might know. So I think we're close to that.
Is it useful to have sort of rather than a hypothetical, what can you do? Maybe an investigator with a very specific question they want to pursue, bring that to you, and then let you make the call as to whether or not that should, be referred to consortium context or something.
Yeah, I'm happy to discuss, and there are obviously other faculty as well. Christian Hume as well. My wife although she does a lot more cardiovascular disease, is also certainly involved in a lot of these studies. But yeah, I think that would be the best thing is if someone's interested in something, we set up a time to talk, and then we can figure out how to best pursue it.
Okay, and just practically speaking, Nikita had a slide earlier about how to reach out to you guys. In just so you don't we don't flood your inboxes. Is there a formal sort of actually not formal, informal way to reach out to you guys individually, or should it go through the TIS, or what's the preferred way to have that kind of consult?
So I think, and Nikita and Nick can chime in on this. What Nikita was describing is for individuals who want to gain direct access to the biobank data, and we have to link the biobank genetic data with the data, the EHR data that's in Compass, and that's a whole process, right? So that's the data access thing, and then there's the idea of, well, I have a hypothesis that's interesting, but do I want to bounce it off someone to see if my idea is any good? I don't think we really have that mechanism directly set up. I mean, that's kind of more of an informal email thing at the point, but that's a good point, is maybe there's we should discuss some sort of central location where someone can draw out a question and we can, figure out who might be best in our center to sort of help take a stab at it.
I think that would be really helpful. I mean, I've sort of gone through this myself personally here. I was really interested in talking to you guys, but I didn't know who to talk to, and I just sent a lot of emails and pestered a lot of people, probably not very efficiently. If you had one kind of portal where you could triage and send the questions to the appropriate investigator, that would be really neat.
So I'm Nikita, and Nick, I'm gonna make sure that there's not such a thing that already exists. I'm assuming.
It doesn't exist yet, but I know Brad Peterson, our director for innovations in CCPM, is working on this.
Okay.
You may recall, Eden, there have been talk about creating those portfolios for the faculty within BPM and CCPM, which sort of summarizes what expertise we have: prostate cancer, Eden Lange, thyroid cancer, Nikita Pozdeyev, and so on. But I don't think it exists yet.
All right. So
and to answer your question, correct? Yes, absolutely. If you have a hypothesis you want to discuss with Eden or with Chris, there is no need to go through TIS. We are a service. Once you talk to Eden and you want the data in the biobank, we help to deliver that data to you.
Got it. So we will at some point have the Ask the Geneticist hotline, but for now, Ethan and Chris Gino are the go-tos. Is that correct?
Yeah, I'm thinking that's. I'm trying to figure out who else might. I'll email you if I can think of some other people as well.
Okay. Jim Costello, you had a bunch of questions. Do you want to jump in here?
I mean, I think most of them have been asked. I've just been thinking about the applications in cancer, obviously, and then you mentioned colonial hematopoiesis, and I think given the depth of sequencing and that it's only from one sample, I think some of those questions are going to be difficult to ask. So I don't know if I have a whole lot to add to that, Craig, other than just kind of getting a sense of what data is available and then what questions we can actually ask of those data, given the constraints.
You raise a good point about the clonal hematopoiesis, Jim. What kind of depth do you think you need to do that?
Depends on what frequency allele you're looking for, right? So at 20x, I mean, unless it's basically heterozygous through the population, I don't know if you would be able to detect much more than that. And then without samples in sequence, right, from the same patient over time, then that limits what you can actually say about it, right? Okay. So, yeah, anyway, I just thinking about what you could ask. I think Evelyn had a good point as well. So, we do have a higher Hispanic population. So is that not reflected in the data that we have?
Certainly not reflected at 22%. Currently for the first 34,000, but hopefully if we recruit over time, we can boost our numbers to get closer to that number.
Evelyn, I'm not sure if you're still on, but is that an issue of effectively recruiting those patients, or are we just not, are we falling down somewhere else?
Yes, that's what I was writing on the chat. Yeah, the problem is UC Health also doesn't have a large Hispanic population. So I remember discussing with Chris, working with Denver Health and other partners to increase representation because they do have larger numbers. So it's a complex problem in that, we need the numbers, but we need the patients. We do have them in Colorado. We just need to increase representation.
Yeah. Other questions for our panel? Okay, well, again, I'd like to thank our colleagues from the CCPM. Really appreciate your time today, and I wish everybody a good afternoon.
Thank you very much for the opportunity.
Thanks for having us.